Hello, welcome everyone. Thank you for joining us today. I am Tony Graham. I'm a member of the class of 1981, also the proud parent of two U of R graduates from the classes of 2015 and 2018. I'm glad you could all join us today. I am also a member of the Alumni Board. This is a group of 25 members representing each of the university schools. Its purpose is to engage all alumni and students in a lifelong connection with both one another and with the university. Today, I'm very pleased to announce that today's lecture is part of a series called Experience Rochester. This exemplifies the university's commitment to lifelong learning and features topics and speakers unique to the university. We have a great number and wide range of registrants with us today. I think we've got over 1,000 registrants. We have those dating back, I believe, as far as class of 1946, all the way to today's students. I also want to welcome those who are going to be viewing the session on YouTube and on Facebook. We have a couple of tips before today's webinar begins. These are Zoom tips, which I'm sure everyone is getting familiar with. A, if you would like to ask a question, please submit it through the Q&A function that is located at the bottom of the screen. B, if you would like to view this session with closed captions, click on the CC button on the bottom toolbar and then select Turn on Subtitles. And if you are having trouble viewing the webinar, we have a phone number that was included with the email, and you can listen in if you are having trouble with the video. With that said, I would like now to introduce today's lecture. Today, we are joined by a panel of experts from the university's medical center who are doing groundbreaking work to help us treat, track, and prevent COVID-19 from spreading. Steve Dewhurst, Vice Dean for Research of the School of Medicine and Dentistry, will moderate a session and will introduce our participants. With that, I want to thank you all once again for joining us, and I'll turn it over now to Steve. Thank you, Steve.
Hi, thanks very much, Tony. Welcome everybody. I can see the little clicker at the bottom. We're still having participants join us, but we're around 460 people. So that's really terrific. And we're really pleased to have so many people joining us today. We have a fantastic panel of really great experts. That's not me, that's everybody else. I'm the chair of the micro department in addition to my other roles that Tony mentioned. So this pandemic is very much sort of in the wheelhouse of what my department studies, but also what we've studied for decades in the infectious diseases division here at the U of R and in pediatrics as well. So as many of you may be familiar as alumni and friends of the university, we have a long history in infectious disease research that's really been immensely impactful and successful. That includes the development of the Haemophilus influenza B vaccine that really led to the virtual elimination of a major cause of childhood meningitis, a disease that was prevalent 30, 40 years ago is essentially gone. And that's thanks to the work of University of Rochester scientists. It's also true that the human papillomavirus vaccine has its roots here, and that's the first vaccine against cancer. And that again is really making an enormous change at the global level, protecting women's health across the globe. So those are examples of vaccine technologies that have been developed here, very germane to the current situation where we're talking about a global pandemic of a novel virus, this novel SARS-2 coronavirus. So this is, as many of you know, it's an RNA virus, and we'll be talking a lot about interventions to prevent the spread and disease of this virus, to look at how are we going to develop a vaccine as quickly as possible, how is the university being part of that effort, how do we develop effective antiviral drugs. So the U of R has already been part of the first trial that led to the emergency licensing of remdesivir. And that's the first drug that shows positive effect against SARS-CoV-2. So that's an example of an impact that we're already having. You'll hear from many of the experts on the call with me today, but just to give you some context, Anne Falsey and Angela Branch direct our vaccine and treatments evaluation unit. That's one of only 9 such units in the country. And they are at the vanguard of developing interventions to prevent COVID-19 disease and to prevent infection with the SARS coronavirus. And they'll speak with you about some of the work that they're doing and some of the plans that they have. Dave Toppin's joining us also. He's from our New York Center of Excellence in Influenza Vaccine or Influenza Research. And obviously there's a lot of parallels here between influenza, another common respiratory pathogen, and the SARS coronavirus. And that whole network that Dave's part of, it's one of only 5 national centers of excellence on influenza. That whole network is being repurposed to study COVID-19. We have another network that's very involved also in the infectious disease division. That's our clinical trials unit, really focused on HIV. But again, given the nature of this global pandemic, that whole research team is being repurposed nationally to focus on COVID-19 interventions. And then Nana Bennett will be joining us, and she's involved with the New York State Emerging Infections Program. And Nana's really an expert in public health and community partnering. And as we think about COVID-19, that's really one of the most important aspects. Dave and Angela will speak with you about where are we going in terms of new treatments and new vaccines. But one of the things we really have to think about is what happens when we get a vaccine. How do we make sure that everybody who needs that vaccine is able to receive it and wants to receive it? So there's a lot of education, trust building, outreach that needs to happen. And we've really been enormously successful over time in doing that. Nana has been at the vanguard of leading efforts to make sure that our community has had a very strong uptake of the influenza vaccine in previous years across all communities, and particularly across diverse communities of color, because those are the communities in particular that are most affected by this COVID pandemic in our country. So there's a lot, I think, of really important, exciting work that the university is leading, and it's really a privilege to be involved with an institution that is playing really a major national role. And maybe one last thing I'll say is that you'll hear, I think, in some of the comments that Anne and Angela will make about a new vaccine that they'll be testing in a phase 3 trial. That's a large efficacy trial. And very likely the lead candidate that we'll be testing is a candidate that also, in fact, has some roots back at the university. The leadership of the company that originally developed that vaccine is headed by a member of our infectious disease division previously. So Tom Evans used to lead our ID division, and now he's in a different role, but really promoting a new collaboration with us here at U of R that's very exciting. So with that, I want to start to introduce each of our panelists. I'm going to start with Dr. Angela Branch. She is an assistant professor in the Department of Medicine in the Infectious Diseases Division, and she's co-director of our URMC Vaccine Trials and Evaluation Unit. So Angela.
Thank you. Thank you, Dr. Dewhurst, and thank you, members of the Alumni Association, for inviting me to talk. I want to start by my remarks by speaking to a conversation I had with local media 2 months ago. They really shifted focus to start to ask about research and how research was being pivoted to address this global pandemic. Up to that point, all the things that you were seeing on the media really had to do with containment, as it should when you're talking about a major public health. So the local reporter asked me, what will research do? How will research be changed or enacted to help in this pandemic? And I said it was really going to be a 3-pronged approach. First, we were going to try to understand COVID-19 as a disease, where communities it's affecting, how it's affecting the body, something we call pathogenesis, how the body is defending itself against it. And that understanding would be needed in order to really inform treatment and vaccines. And then the second sort of arm of the research efforts is to really start to develop ways of treating COVID-19. When we started, we had no proven treatments that would be beneficial. And we were really working very much in the dark with that. And then lastly, one of the most important efforts would be to find ways to prevent the spread of infection, and that's what we do with vaccine work. So for Ann Falsey and I, as Dr. Dewhurst mentioned, we are co-principal investigators of the University of Rochester Vaccine and Treatment Evaluation Unit. And that story actually starts a little bit more than a year ago, when Ann and I and Steve and Dave sat down and talked about this, and we decided that this would be a program we were interested in bringing back to the university. It's the oldest network of the NIH. It's been around for some 60 years, and this is what they do. They develop vaccines and treatments for emerging public threats that are infectious. So we were really excited to be part of that, and we were notified that we were awarded the grant from the NIH to start that unit on the 12th, I think, of December. This was before we knew anything about COVID-19. On December 19th, something gets published coming out of Wuhan, China, that there's this new pathogen that's causing severe acute respiratory syndromes, very similar to what we've seen before. And then by early January, we had a disease and a name to that virus. On January 21st, Ann and I and Dave were at the NIH in a meeting, our first inaugural meeting of the VTUs as a leadership group. And Anthony Fauci comes in the room and he says, have you guys heard about this coronavirus that's coming out of China? This is your mission. This was what we expect you to work on first. So mobilize, go home and mobilize your teams and get ready to start working on treatments and vaccines for COVID-19. That was one day after we had our first case here on US soil. So everything since then has been moving very rapidly. I'm going to talk a little bit about what our first task was as a VTU, which was to work with developing a therapeutic agent. We started to see local cases here in Rochester in March. And by that point, we had developed within the VTU and the leadership group a protocol to test a drug called remdesivir. Remdesivir is an RNase polymerase inhibitor, and what that means is that it stops the virus from making more copies of itself in the body. And it was actually developed for Ebola, but it had been tested against other coronaviruses, and the network and the NIH felt that this would be sort of the most promising thing to study first and to study quickly. We started that trial in mid-March. By the end of April, the trial was fully enrolled. We enrolled 1,040 subjects all across the world. 16 of them were here at the University of Rochester. And we gave half of those people remdesivir, and we gave half of them placebo. It was really interesting for Ann and I to perform that study here at the University of Rochester because in those first early days, walking through the highly infectious disease unit, the ICU, down on the 3rd and 4th floors, and seeing just really, really sick men and women who were struggling to breathe and on ventilators, there was just this tremendously enormous pressure to want to do something. And we kept having to remind ourselves that we don't know that this drug works. And so not to give people false hope and false expectation, but when you're dealing with an enemy that you don't really understand yet, and people are as sick as they are, there is a temptation that doing something is better than doing nothing. And so, you know, sort of weighing that and really trying to do what we can to offer benefit to people. And then finding out, you know, within the last month that in fact remdesivir has been shown to have therapeutic benefit in helping people to recover and even decreasing mortality. In the studies, I think something that I'm starting to appreciate is that they looked at mortality benefit in folks who got remdesivir and those who didn't. And it didn't reach what we call statistical significance, a difference in the number of people that died who got the drug versus those who got placebo. But it almost did. And in fact, it was an 8 to 11% difference. And when you think about 100,000 people in the United States dying from COVID-19 within the last few months, that's 3,000 lives that potentially could have been impacted by this drug. And so we are enormously gratified to have been part of this study to help. We saw some remarkable recoveries of people that we know got drug. And we saw that it's probably not good enough. So that trial was called ACT, the adaptive COVID-19 treatment trial. That was Act 1. We're currently in Act 2. Remdesivir has become sort of the standard of care, and we've added a second drug to it called baricitinib. This drug is a potent anti-inflammatory in that it inhibits a potent mediator of inflammation. And as many of you may know, COVID-19, one of the problems is not just the virus, but the system-wide inflammation that's being triggered by this infection. And so we think that the combination of remdesivir and this second inflammatory inhibitor might be better than remdesivir alone. As I said, you know, the results of the remdesivir trial are incredibly just encouraging, but we know that we're not done. So we're in Act 2, and there'll likely be an Act 3 and possibly even Act 4, and we think we can do better, and that's what we're working towards.
Thank you very much, Angela. That's a great overview. Our next speaker that's going to be joining us is Dr. Anne Falsey. Anne is professor in the Department of Medicine, also in the Infectious Disease Division, and she too is co-director of the VTEU. So Anne?
Hi,
Hey.
thank you, Steve, and thanks to the alumni for joining us in this webinar. And thanks to Angela there, she set me up very nicely explaining who we are and what we do. And I've been asked to sort of update what the University of Rochester is doing as far as leading the way locally for vaccine development. And what I thought I would do is begin with a little bit about vaccine development and how what we're doing fits into the grand scheme of things. So many of you who are medical people will know that there are different phases of vaccine development, phase 1, 2, and 3. Phase 1 is generally small studies focused on safety and immunogenicity to see if the vaccine is tolerated well and produces an antibody response. Phase 2, we start to expand to look for doing dose ranging and finding just the right product to bring forth into very large vaccine efficacy trials, which are referred to as phase 3. There are a number of trials. They are both pharmaceutical sponsored and sponsored through the NIH networks, and many different approaches. I looked at the websites just recently, and there's about 28 companies actively with programs both ranging from preclinical, where they're just testing it in animal models, to ones that are fairly advanced. They range from RNA vaccines, DNA vaccines, recombinant, adenovirus chimpanzee vaccines, DNA, traditional protein-based vaccines. Most of the vaccines currently are focused on developing antibody to the spike protein, which is one of the major surface proteins of the coronavirus. And I'm sure that everybody appreciates that an effective vaccine is really the only way we can all get out of our houses eventually, that we need to prevent this disease. It needs to be a combination of therapeutics and vaccines, because no vaccine, well, almost no vaccine is 100% effective. And I'm sure that many people on the webinar have heard about Project Warp Speed, and that is an interesting government program where we are pushing through these phases that I mentioned very rapidly. I think it's important to say that nobody is sacrificing safety, that phase 1 is always part of any vaccine development plan. But what is unusual is that manufacturers are scaling up vaccine production ahead of knowing whether or not they actually have an effective vaccine. And normally that is never done because you could be wasting a huge amount of time and energy making a vaccine. But clearly, if we end up at the, in the fall with studies that show we have an effective vaccine, it's a great advantage to be ready to put that vaccine into the population. So currently, the University of Rochester and the VTEUs are part of a new network which has been put together partly for Project Warp Speed, and it's called the CoVPN. And it is it takes advantage of existing networks. So the VTUs have 9 sites in the US, but we are partnering up with our colleagues who have been traditionally doing HIV vaccine work. And so those are the ACTGs, the HPTNs, and the HVTNs. And so locally here at the University of Rochester, Mike Kiefer has led the effort with the HVTNs. And so we will be participating in these network studies. And one of the big advantages of having this giant consortium, and it's really kind of unique, is that the major manufacturers are moving their products through the networks. So the first one is Moderna, which you probably have heard about. It's an RNA vaccine. We will be participating in August with the AstraZeneca product, which is what Steve mentioned earlier, which was in part developed with Tom Evans, one of our previous ID faculty members. And that is the adenovirus, chimpanzee adenovirus vectored vaccine that is in trials presently in the UK. But shortly after that, there will be a number of other vaccine companies that are lining up to do very large efficacy trials. So here locally in Rochester, we did begin a vaccine trial with Pfizer. It was phase 1. It's a joint University of Rochester and Rochester Regional Health effort. And some of the folks on the call may have called to volunteer. And that's really great. We need young, healthy people and healthy older people to participate in phase 1 trials. But as we move through the trial, the phases, we will be reaching out to many members of the community and high-risk people with underlying conditions, as we start recruiting for the efficacy trials. And as mentioned, it is very important that we get people from diverse communities, people of color to volunteer for these trials, since we want to make sure that we have a vaccine that is good for everybody. So that's where I'll stop. We are, laying the groundwork for some very, very big vaccine trials, and it will be truly a community effort. Thank you.
Thanks very much, Anne. One thing I should add, both Anne and Angela are directly involved in patient care in this crisis. So it's a very unique perspective that they both bring, both a research perspective and direct care. So anyway, want to next introduce Dr. Nana Bennett. Nana is a professor of medicine and public health sciences. She's also the founder and director of our Center for Community Health and Prevention, and she is the co-director of our Clinical and Translational Science Institute. Nana?
Thank you, Steve. And thank you to all of you for joining us today. It's really a pleasure to discuss this very complicated health issue with you and to talk about all different aspects of how the university is approaching COVID-19. As Steve mentioned, we in the Center for Community Health and Prevention try to marry research to community service. And that means that we do a lot of work with community partners. We try to really bring the voices of the community into the medical center and also take the expertise and resources of the medical center out into the community. As soon as this crisis started in Rochester, we knew that we would need to step up and to work with the community very closely. That meant initially working with the local health department to really track the epidemic in our community. We actually help them to collect data. We help with a lot of their analysis of data, and we are in constant communication with them to ensure that their messages are getting out to the whole community. That second part of talking to the community is part of our community engagement mission that we have been doing for many, many years, but it's taken on a new urgency with COVID. And we've worked very, very closely with our community partners to try to understand what they need in this time and whether we can help them to get what they need. We've also worked with them very closely to figure out how to take public health messaging and make it relevant to diverse communities. Sometimes the messaging that's the formal messages that come down from the national level are really not very well adapted to our community. So we've worked very closely with a number of community agencies to adapt those messages and make them work for our community so that we can prevent COVID-19 from spreading. The third area that we've been working in is research. And we have been part of a network called the Emerging Infections Program for more than 20 years. It's a Center for Disease Control funded program that tracks infectious disease across the country by doing population-based assessment and surveillance. And like many of the other networks you've heard about today, as soon as COVID-19 happened, that network was transformed into something called COVIDNet. And COVIDNet is the source of a great deal of population-based information about the burden of disease produced by COVID-19. We have been tracking every hospitalization in our region and collecting fairly in-depth data about those hospitalizations. The purpose of this is not only to determine the burden of disease in our population, but also to see what we can learn about the clinical course of disease, the impact on future care, the death rate, both in hospital and out of hospital, and really define what's happening as a result of COVID-19. The second thing that's very important about this network is we try to identify the populations who are at greatest risk. And as everyone I'm sure on this call knows, it's been very clear that populations that suffer from the social determinants of health impact have been very severely impacted by COVID-19. But we've tried to look at those data in more depth and really understand why that's happening. Is it a problem of healthcare, or is it a problem of actually contracting the disease? And in our community, we've found that it's the latter. It's really the exposure and the risk of contracting the disease that has determined the clinical course for populations at especially high risk. We also look at other risk factors, and I think everyone knows that age and the impact of chronic disease has been very clear in this epidemic, but we try to get a lot more granular about that and try to understand the impact. The last really useful thing about having this network is that it's a platform for assessing the effectiveness of vaccine once we have a vaccine. So while we do clinical trials to understand whether or not a vaccine works, we actually need to use the vaccine in a population to determine whether it really reduces the burden of disease. So this network provides a tremendous platform for looking forward and seeing whether or not we can really reduce the burden of disease of COVID-19. So we've tried over the course of the last several months to really combine and focus on our service and on our research. And we hope that that will create a level of trust and information in our community that will make all of our studies easier to complete. And we work very closely with all of our investigators across the institution to try to make this happen and to ensure that we can recruit diverse populations and really understand the impact of our interventions. So thank you.
Thanks very much, Nana. And our final panelist is Dave Topham, Dr. Dave Topham. He's the Marie Curran Wilson and Joseph Chamberlain Wilson Professor in the Department of Microbiology and Immunology in the Center for Vaccine Biology and Immunology. He's also the director of our New York Center of Excellence, Influenza Center of Excellence, and the new director of our established brand new Center for Coronavirus Research. So Dave will tell us a little bit more about that. Dave.
Good afternoon, everyone. Yeah, so I'm the lone PhD here, I guess in addition to Steve, but I'm trained as an immunologist, known best for studying influenza, but also other respiratory pathogens. And so we've got several different studies ongoing to try to understand how the immune system responds to this virus and eventually how it responds to vaccination. One of our first studies is to look for pre-existing immunity in people that have not yet been exposed to the virus as a means to find out whether or not we can determine populations who may be at more or less risk of infection and disease. The second study that we've got ongoing is looking at people with acute infection to try to understand correlates of severity with the immune response, and also to gather information about, what does a successful immune response against this virus look like? Because we can use that information to inform whether or not our vaccines are producing the same kind of response. Ideally, you want a vaccine to essentially give you the beneficial response that a successful response to infection does. And we're doing these studies here in Rochester, but we recently got funded to do surveillance studies for COVID-19 and influenza in the Southern Hemisphere. This was part of the SEERS network. So 5 centers got together and put in a single proposal. We have sites in Australia, all 3 major cities, and in Vietnam. And so we will be conducting surveillance for these viruses and studying immune responses over the course of their flu season, which of course is during our summer. So combined, we're going to be gathering a lot of samples, a lot of information, and we should have a pretty good handle on how the human immune system responds to these viruses. Now, I'll say a little bit about the Center for Coronavirus Research. So, you're talking to a small number of us here today who are doing coronavirus research, but really there's many investigations going on in different labs across the university, not only in the School of Medicine, but also on the River campus in arts, sciences, and engineering. And so as a way to coordinate all these different efforts and look for ways we can develop synergies among our researchers, we thought it'd be best to have a single center that can be a resource that researchers as well as the public and our alumni can go to find out more about what research is ongoing, and again, try to hook up investigators who might benefit from each other's knowledge and technologies. And that is literally just started last week. So we're hoping it's going to be a tremendous success.
Thank you very much, Dave. So maybe just to jump on some of the things that Dave was alluding to, one of the things that we really need to do in terms of making our coronavirus research more effective and better integrated with the community is figuring out, for example, how to reach out into the community to do what will be unprecedentedly large vaccine trials and multiple vaccine trials. There'll be as many as 5 new vaccines being tested through the networks that Anne and Angela are intimately involved with. And we'll be enrolling probably 1,000 people at high risk for acquiring COVID right here in Rochester in the next couple of months. There's all sorts of challenges with that and things we need to put in place. That includes, for example, the research pharmacy. How do you make that number of doses of a vaccine quickly? How do you make sure they're provided out? It includes how do we reach out into the community? So, for example, we're exploring right now, do we need to get a bus, a mobile unit, and get out into the community? Nana's smiling because she's been very involved, and the CTSA has been pushing that for quite a while. And all of a sudden, it's become really an immensely important need. But there's a lead time with getting a bus. Outfitting a bus so that you can do studies in it is not trivial. What do we do in the interim? So there's lots of sort of brainstorming going on right now to try and problem solve a whole variety of issues. One of the things that came up on the chat and I think is really central to this whole discussion is, do we know that immunity to COVID will confer long-lasting protection? It's a super important question, and it's at the heart of, how likely is it that we'll get a vaccine? So the folks on the call are by far the best qualified people, I think, at the university and even nationally to answer that. One thing I will say is that there are some studies that are going to be ongoing that will give an indication. And those are studies using monoclonal antibodies, either as prophylaxis or as therapy. Prophylaxis meaning prevention. And I guess maybe I'll throw the question to the whole panel. What is your sense of previous infection with SARS-CoV-2? Is it likely to confer durable immunity against future COVID-19?
So, Steve, that's a really important question, and we're just starting to see data emerge on that. It looks like infection does produce sterilizing immunity. The immunity persists for some period of time, but we're not quite sure how long. And that there's now evidence that people who have been infected once can get infected a second time. However, those people that got infected the second time, and this is mostly from healthcare workers, they don't shed the virus, and the virus is present at very low titers. So they're not going to spread the virus around. But it does say that the immunity is not perfect. And so again, once we have vaccines out there, we're going to have to watch very carefully with Nana's network to make sure that even though a vaccine elicits an immune response, that it's actually a protective one.
And just to jump on what you said, Dave, if I understood you, you're also suggesting, I think, that those healthcare workers, presumably quite rare, that got a second infection, that wasn't symptomatic severe COVID disease.
Correct.
It was mild, right? Yeah, yeah.
Correct. They had
so
no
that would
symptoms,
suggest,
and yeah.
yeah.
So I could add to what Dave said. This is such a new virus. We're not very long into it. So making predictions about immunity is rather challenging, and differentiating true reinfection from persistent detection of RNA is another challenge that we have. But the main thing I wanted to say is that I can't imagine this will be that much different than the other respiratory viruses. We do have some experience with coronaviruses, but there really is no long-lasting protection to any of the respiratory viruses. If you think about it, we all get infected in childhood with these viruses, and there is some immunity for a period of time, generally a year or two, but we do get reinfected with these viruses throughout our life, because serum antibody wanes, nasal antibody wanes, and generally the infections are not as severe. And so I would predict, and it's with, some caution, because again, we're very new into this, is that, for a year or two, you will be relatively protected. And then after that, you will become susceptible again. But that if you have some antibody and you can have an amnestic response, that it generally should be a less severe infection. I would be surprised if people were susceptible to widespread pneumonia and the kind of very severe disease that we're seeing. But again, because we don't have experience, you can't say that with certainty.
Wanted to take one of the questions that came into the chat was from Joe, who was asking a question really about the virus changing, because obviously with influenza, that's a real challenge, as Nana in particular is very familiar. My sense, and before you all answer, my sense that the SARS coronavirus is a much less, it has less ability to generate completely novel variants perhaps than flu does. There are some important differences genetically between the viruses, and its polymerase is less error prone and has what's called proofreading activity, which means it spits out fewer mutations. But nevertheless, the question about whether the virus might change over time is a very reasonable question. And maybe Nana and then Angela, could you maybe comment on that?
Yeah, I'm probably not the best person to comment on it, but I have studied influenza for the last 30 years. And certainly what we see at a population level with influenza is very complex, as I'm sure everyone knows. And that's the reason that we have to be vaccinated every year to have protection, because influenza does mutate quickly. I don't think there's, my sense is the same as yours, Steve, that this virus may not be the same as that, but there's still the question that Anne raised as to how long will immunity last. So is this a virus for which we will have to be vaccinated every year, I think is a reasonable question, or every 2 years, or something to maintain immunity. But we're certainly hopeful that we won't see the same complex process that we have to go through with influenza every single year, where we have to identify the circulating strains and develop essentially a new vaccine every year. That would be less likely, I think, with this virus. And we can certainly hope that it's less likely.
Thanks.
Go ahead. I would agree with Nana. I think it probably won't be like flu in the sense that you have these various different ways that the genes can reassort, as is true with influenza, which is why we tend to see new strains popping out every few years or so. I think maybe, as you said, the mutation rate and even the types of mutations are going to be less facile with COVID-19 or the SARS coronavirus 2. And it's possible also that as the virus mutates, it actually attenuates itself and becomes sort of less pathogenic than the wild type virus, meaning causing less severe disease. So that would be a mutation that we would welcome if it turns out that this coronavirus becomes like all the other ones and just causes mild colds throughout the winter seasons.
Yeah, and Steve, my lab has developed a technique where we passage viruses in the presence of immune sera. And what you see at the end of that, at least with influenza, is you see mutations that mimic what actually happens out in the field in terms of what parts of the virus change under that artificial immune pressure. We're conducting those experiments right now. Again, I'm sort of expecting not to see a whole lot of change because coronaviruses are known to be fairly stable, as you mentioned. But we want to do the experiment anyway.
Yep, thanks. There's a question from David asking if one wants to volunteer for vaccine testing, how do you participate? That's probably something we should be sending out a little extra info from our friends in advancement, but I didn't know if any of the folks on the panel wanted to respond to that too. So, because I think it's probably many people on the call would be interested in participating in some way, and how do they best do that?
Angela, can you describe the landing page?
Sure. So as Anne mentioned, we're most likely going to be launching a large phase 3 vaccine study towards the end of July. That means we have to actually start recruiting subjects for that now. And it will involve, between our network and the HVTN that Mike Kiefer is leading, about 1,000 subjects here in Rochester is what we think. And so we really would like as many people to volunteer, healthy people, people who may have some underlying medical conditions, but they're very well controlled. We are working with the University of Rochester media team to develop a landing page, and it's covid19research.rochester.edu. If you go to that landing page, it has a whole list of questions for folks that are interested in participating in research related to COVID-19, which spans the spectrum of everything for acutely infected, people who've recovered from infection, and people who are healthy but interested in participating in vaccines. There'll also be a lot of things on social media, at Rochester, on the URMC website. We'll probably be doing news stories that will give you links as well as phone numbers on how to volunteer. So stay tuned for that. It will be very widely disseminated, I'm sure.
So if I could just make a comment, that we need different kinds of volunteers for different phases of studies. And while we wish that nobody was at risk for getting coronavirus, when we get into the big phase 3 efficacy studies where we're going to be recruiting 1,000 people, we particularly would want to enroll people who are, unfortunately, by what they do or where they live, at risk for contracting the virus. So we will be reaching out to first responders, police, healthcare workers, potentially teachers if they open the schools, daycare workers, retail personnel who, by the nature of their jobs, they come into contact with the public. So just to keep that in mind for anybody listening, if you fit into one of those categories. Thank you.
Yeah, and I just wanted to also accentuate that there are a lot of studies going on at different stages. So if you, for example, have been exposed to COVID-19 and are interested in entering a study, or if you have COVID-19 and have mild symptoms, or if you are in the hospital and have COVID-19, there are studies at every stage along the continuum, and we are actively recruiting for some of those studies.
So that relates a little bit to a question about asks for the alumni network. So certainly one of those asks would be that, you know, if you're interested in participating in a study, please do go online and find out more. There are other ways that you can help us. I sort of alluded to some of the things that we need to start thinking about doing, like, you know, getting a bus or getting, we may need to expand our trial capacity, which means, you know, we can't throw up a building in a matter of months, but we can buy or rent a mobile classroom, or, well, it wouldn't be a classroom, it would be a bunch of exam rooms, but those kinds of things. And you can imagine those are not cheap financially. So also financial assistance is part of what would be helpful, just to answer that question directly. There's a question about how many doses of vaccine would be required. I would say in general, and anybody else should correct me, but I think the networks that you heard, these alphabet soup of, you know, the VTEU, and then there's a whole bunch of other ones. The networks are choosing vaccines where they're confident, I think, that production and scalability are going to be possible. So there's not really that much point testing a vaccine for efficacy at a phase 3 trial if we don't think we can scale that up to millions and millions and millions of doses, because obviously it's a global pandemic. We in fact will need to make not just millions of doses, we'll need billions, and we'll need to get them rather quickly. So I think that's certainly part of the consideration about what goes in. There was a question, actually a couple of questions that relate to some of the aspects of how COVID-19 disease presents. So in many people, it can present with cardiovascular symptoms, it can present with neurologic symptoms, it can present with thrombosis or stroke. And I guess maybe Anne and Angela in particular, because you see patients with COVID, just curious and your responses to that. Angela, do you want to, or
yeah, Angela, you go ahead.
Well, as you know, when I spoke about our treatment trials, I mentioned that we know that an antiviral alone is not necessarily effective for people at certain stages of diseases, because at that point, it's really not just trying to stop the virus from replicating. It's sort of this system-wide inflammation that's been triggered, which once it turns on, it takes a long time for it to turn off, regardless of whether or not the virus is still replicating in the body. And it's this inflammation that's really leading to some of these different things that we're seeing. I think the inflammatory process is affecting the heart. We see it affecting the liver. We see it affecting the kidneys. Certainly we see it affecting the vasculature, and that's why people are developing clots, both clots leading to strokes in the brain, but also clots in the extremities and clots leading to clots in the lungs. And I think that figuring out a way therapeutically to intervene at that part of it and stop and turning off that inflammation is really a major priority for our network. And I think for anyone who's sort of seeing patients and involved with their care. In the ICU, they're doing lots of different things that we know to do for patients who are inflamed from other things, other viruses. But we're finding that while that's partially effective, certainly it's not the whole thing, the whole of what needs to be addressed. Like
thanks very much.
Yeah.
You know, I would just echo what Angela said, that, you know, at the bedside, we're observing certain things, and we're just so new at this. This is a new disease, and it reminds me, I'm old enough, of observing when HIV first came through. And we have so much to learn. We're making observations about strokes and inflammation and, you know, different biomarkers, but how it all fits together to produce the syndrome is going to require some significant research. So we're working with our immunology colleagues, we're working with Dr. Mariani and looking at functional genomics, which genes are turning on and off. But, you know, I think that what I'm struck by is how much we need to learn to understand how to better intervene and then, you know, even protect.
Thank you. So just to try and keep us on schedule, I'm going to already, I'm afraid, have to move to final comments from each of our panelists. And I'll just preface that by saying I'm very appreciative of all the comments we have in the Q&A. It's really terrific We will try to answer some of those offline. Obviously, we aren't able to get to all of them right now. So in terms of closing comments, Dave, if you maybe wanted to start us off.
Yeah, I welcome the support of our alumni. As we've all just mentioned, we have a lot going on here. It can be a little overwhelming at times, but I think we're managing. But I think we can also have significant impact on a local, national, and even global scale. And so I was telling somebody the other day, it's both an exciting time because as a researcher, we like to study new things, but it's also very anxiety provoking because there's so much pressure and you want to do well. So we'll do the best we can.
Thanks, Dave. Nana?
Yeah, I can only agree with all of my fellow panelists that this is a time of tremendous learning for all of us. And I think for communities at large, it's a time of tremendous learning. And I think that we could not have predicted exactly how this pandemic would play out in our communities. And we're seeing it change every single day. And I think the really wonderful thing is that it's made people realize we're all in this together, and also that science is critical to our moving forward. There's a lot of debate about science, anti-science, whatever. I think that in all honesty, this epidemic puts those debates down and really shows us that we have to have good science and we have to support science, and that science is key to our survival. And I just am really happy that the university has created an opportunity for us to talk to alumni and community members about science and why we think this is such a critical endeavor at a time of such huge uncertainty.
Thank you. Anne?
Yes, I would echo again some of the comments that I've heard that, I've said to people who comment, it's either a really good time to be a respiratory virologist or a really bad time to be a respiratory virologist. But I'm very, very proud of the work that we and our colleagues at the University of Rochester are doing. And I'm very acutely aware of the gravity of the importance of the work that we're doing. And so I hope people will be patient with us as we work through as much of the science as we can.
Thank you. Anne? Sorry, Angela?
So one of the things that has really been paramount in my mind the last week or so, as Anne and I have discussions as to what's coming this summer, is how much we don't know yet about how this is all going to unfold. Which one of these vaccines will make it to phase 3, will make it through phase 3, will then be entered into populations. What's going to happen with COVID-19 infections in the fall? And in the winter, will we have second surges? Will it go away? Will the virus attenuate? There's so many unknown questions. And the daunting thing is thinking that as hard as the last 3 months have been challenging, I think both professionally and personally for all of us and for our entire community, we have another year of this. So I've been thinking about how you do another year of this. And I still don't know. But I do know that once we're on the other side of this, we'll be really proud of the work we did here at the University of Rochester.
Thank you. I think that's a great point to make, Angela. Just, I think there is a sense of purpose. Dave mentioned that many labs are working on COVID here, and it's a big change. Lots of labs that used to work on something else have switched over to COVID because there's such a desire to make a difference, to contribute in a positive way. And I think, that includes being a volunteer in a vaccine trial. It can include any number of things, but that sense I think we all share of wanting to make a positive difference, have this pandemic be resolved and really save as many lives as we possibly can. Those are fundamentally important. And as Nana said, I think that appreciation that we have for medical research and for science, for education, for understanding, for knowledge, those are, I think, some of the really important lessons, that the lessons about what do we think about another pandemic in the future, because it's pretty clear that if COVID can happen, it could happen again, and we need to be better prepared going forward. And I think that's one of the other lessons that I think we'll be taking away from this pandemic. So I think with that, maybe I'll turn it back to Tony for some closing remarks. Tony? And it's only Tony's camera.
Thank you very much, Steve. Thanks to two groups here. First of all, to the panelists. It was a wonderful and informative session. As someone said in the questions, you rocked, so well done. Very, very enjoyable. Second, to all the participants here, great questions. I was reading them as we were going along, and really just great to have such an engaged and knowledgeable audience. So thank you all panelists as well as registrants. If you would like to learn more about the Center for Coronavirus Research, there's going to be a link, and it's coming up right now, I believe, that you can click on, and that will redirect you to a site where you can learn more about the Center for Coronavirus Research. Finally, today's program is only one in a series. We've got the next version of the Experience Rochester webinars on June 18th. That's Thursday, June 18th, also at 12 o'clock. This almost seems like a nice segue into that webinar, and that's going to be on COVID-19 and the brain. It's going to feature two professors, John Fox and Steve Silverstein. So hope you all will sign up for that. I think if you enjoyed today, you're certainly going to enjoy that one. That's it. I think we're right on time. So thank you all. And thank you once again to the panelists. So long.
Thank you very much, Tony.